Post-transcriptional modifications, the splicing mechanism. This splicing mechanism is the second RNA processing event that takes place in a eukaryotic cell. And the essence of the splicing mechanism is that a primary transcript that is formed is converted or is through splicing made into a functional mRNA, which then can go in for translation. So let us look at the learning outcomes of this session. So splicing, as mentioned, is the second processing event that takes place in the eukaryotes. Most genes in the eukaryotes have interspersed within their coding regions, and these coding regions are called as exons. There are certain non-coding regions as well present, which are termed as introns. And hence, the primary transcript that is formed by the RNA polymerase will have both the coding regions as well as the non-coding region, forming what is called as mosaics. These non-coding regions, that is the introns, can be spliced out to give rise to the functional mRNA, and the splicing mechanism is aided by spliceosome machinery or by self-splicing introns. The number of introns in genes can vary anywhere from 1 to hundreds, making the gene much larger than its final transcript size. So now let us look at why splicing is required. So say consider you have a gene, okay? This is the DNA in which you have the promoter region, and the promoter region is followed by the start point, and the start point to the terminus you will find that there are several coding regions, okay, which are interspersed with the non-coding regions. So therefore these coding regions are what are the exon 1, exon 2, exon 3, exon 4, which means this gene has 4 exons and it has 3 introns. So when RNA polymerase, having bound to the promoter, transcribes this gene, it will transcribe the entire gene in such a way that even the RNA transcript, which is the primary transcript, also called as the pre-mRNA, will contain all the exons along with the introns. So the RNA that is formed is also a mosaic that contains the coding regions and that contains the non-coding regions. Of course, towards its 5' end and the 3' end, there are certain sequences which are called as the untranslated regions, and these regions are not translated by the ribosomes. Now, when this undergoes what is called as a second processing event, that is splicing and also poly A tailing and 5' capping, it results in a functional mRNA that has at the 5' end a 7-methyl guanine, which is the 5' cap. You can see that the introns have been removed, spliced out, and all the exons are joined to each other. So exon 1, 2, 3, 4, they are joined together to form what is called as a functional mRNA. So this over here at the 3' end is the poly A tail. So eventually, after the RNA processing events, one gets a processed RNA with introns removed. When all introns are removed, then it is what is called as complete splicing or absolute splicing. Now, how does the splicing mechanism know that in a pre-mRNA, which regions are the coding regions and which regions are the introns or the non-coding regions? So let's look at the pre-mRNA transcript. Say, for example, you have a pre-mRNA transcript that has exon 1 and exon 2. So obviously, this intervening sequence is the intron region. Now, the borders of the exon 1 and the intron and the intron and the exon 2 have to be demarcated or defined. That means the splicing machinery has to know that this is the point at which the cut will have to be made. This is the point at which the cut will have to be made to remove the intron and bring exon 1 and exon 2 together. So the question that comes is, how does the splicing mechanism know that this region is the intron region and this is the border of the exon 1 and intron and this is the border of the exon 2 and the intron? So the answer to this question is that the intron itself suggests that it is an intron, because of certain specific sequences that it has, and these sequences are conserved sequences. So let's say, for example, the intron regions have specifically what is called as the 5' splice site. So at the 5' end of the intron is present what is called as the 5' splice site, and at the 3' end of the intron is present what is called as the 3' splice site. Both 5' and the 3' splice sites are conserved sequences. Other than the 5' and the 3' splice site is also present a region which is called as a branch point site. This branch point site has a sequence, and within the sequence which is conserved, there is a highly conserved A. So the branch point site sequence itself is conserved, but within this conserved sequence is present one A that is highly conserved. It doesn't change at all. And what is also observed is that the branch point site is present more towards the 3' splice site rather than the 5' splice site. So that is how it is positioned. So I repeat, an intron region will have a conserved 5' splice site, will have a conserved branch point site, and will have a conserved 3' splice site. Other than this, it also has, just beyond the branch point site, a region. Okay, before going into that, look at the sequence that is conserved in the 5' splice site. So it is GUA. So it is either A or G, and AGU. So it is basically GUA, AGU, or GUG, AGU. Of this sequence, GU is again highly conserved. While at the 3' splice site, there is a polypyrimidine tract. So there is a sequence of pyrimidines present just beyond the branch point site, followed by the 3' splice site that ends with the sequence AG. And this AG is highly conserved. So you can see that this is highly conserved, this is highly conserved. Both are present at the borders of the exon 1 and the intron. And plus you have the branch point site which is also conserved. So interesting to note is that this being an RNA molecule, the phosphodiester bonds that are formed in the RNA molecule are between the 3' OH and the 5' phosphate. So all along you have the 3', 5' phosphodiester bonds present between the nucleotides. But being an RNA molecule, the sugar would also have a 2' OH, because we know that ribonucleotides essentially are made up of riboses as sugars, and ribose have both at its 3' and 2' carbons OH present. So therefore, this A over here, which is highly conserved, will have a 2' OH that is free. And that 2' OH will act as a nucleophile to carry out the first nucleophilic attack. So the 2' OH will basically cut at the border of the exon 1 and the G of the intron to carry out the first reaction of splicing, which is called as the first transesterification reaction. So I'm repeating what I said. The A that has a 2' OH, that 2' OH will act as a nucleophile and cleave a phosphodiester bond, which is exactly between the exon 1 and the G, that is highly conserved, of the 5' splice site of the intron. So let's consider this in a higher resolution. So for example, these are the nucleotides of the exon 1, and between the exon 1 nucleotide and the G, this is the phosphodiester bond that is going to be attacked by the 2' OH of the A. So we are talking basically about this phosphodiester bond being attacked by the 2' OH. So the 2' OH will cleave this bond, and instead it will form a bond with the G. So effectively what is happening is transesterification. That is, transfer of the ester bond that is present here to the A of the branch point site. I'm repeating what I said. The exon 1 is bound to the 5' splice site of the intron through the ester bond, which is a 3', 5' phosphodiester bond. That 3' 5' phosphodiester bond is cleaved by the 2' OH of the A and in turn the 2' OH will now be an ester bond with the G. So it cuts this ester bond and forms a new ester bond. So just that the existing ester bond is broken and a new ester bond is formed. That is why it is called as transesterification. That is, an ester bond has been transferred from one side to the other. So you basically will have the A of the branch point in a interaction with the G of the 5' splice site of the intron. So let's look at this clearly. So you would have now, because of the transesterification reaction, the first transesterification reaction, the exon 1 with its 3' OH free, because that bond, phosphodiester bond, is broken to leave a 5' phosphate of the G and the 3' OH of the N of the exon. That 5' phosphate of the G is now going to be bound to the 2' OH of the A. So what you have over here is a 2' 5' phosphodiester bond formed between the 5' splice site G and the branch point A. So you can see that the G is coming closer to A, or you can say that the A comes closer to G. So the 5' splice site and the A branch point have to come to proximity if the first transesterification reaction has to be carried out. So what you can see now is very clear. Exon 1 has been removed from the intron, but this intron is still bound to exon 2 through its 3' splice site. So the next reaction that will happen is thanks to the 3' OH of exon 1. So this free 3' OH will act as the next nucleophile and attack the 3' splice site G. So the phosphodiester bond that is present between the N of the exon 2 and G of the 3' splice site will be cleaved by the 3' OH. So you can see this 3' OH of the exon 1 is cutting the ester bond over here to form the ester bond between the exon 2 and exon 1. So now what you have is exon 1 and exon 2 joined together by an ester bond. How does this ester bond formed? By the second transesterification reaction, which means the ester bond between the 3' splice site of the intron is broken with that of the exon 2, and instead a new ester bond is formed between exon 1 and the exon 2, thereby joining the exons together and leaving the intron in the form of what is called as a lariat. So this structure that you see, where the G is circularizing on the A and you have a tail of the 3' splice site remaining, this structure is what is called as the lariat structure, and the intron is released as a lariat structure, which can be degraded. Of course, there have been many studies wherein a lariat intron has been converted to a ribozyme that can have catalytic activity. We will take this up in a later session when we talk about self-splicing introns. So this basically is the overall mechanism of splicing, where two transesterification reactions take place. The first transesterification reaction is actually ensuring that the exon 1 is separated from the intron, while the second transesterification reaction at the 3' splice site of the intron ensures that exon 1 is now in a bond with the exon 2, while the entire intron region is released in the form of a lariat. So this second transesterification reaction leads to the splicing out of the entire intron. Now you can also have a phenomenon which is called as trans-splicing. Now what do you mean by trans-splicing? What you observed here was splicing within the same RNA, right? The RNA has the coding regions and the non-coding region, and the non-coding region is being spliced out by the two transesterification reactions. But you can also have two RNA. So you have RNA1 with its exon 1, and this you can see is the 5' splice site. And you have RNA2, you can see the exon 2, and you can see the 3' splice site with the branch point A. So it is possible that the branch point A of RNA2, that is the intron of RNA2 using its A can carry out a splicing at the 5' splice site of an intron on RNA1, a different RNA. So what happens therefore is that the intron of the RNA1 is removed by the intron of the RNA2. Okay, and in turn this exon, which will now have a 3' OH that is free, can cleave at the exon 2 of RNA2. So effectively exon 1 of RNA1 can link with the exon 2 of RNA2, right? That is why it is called as trans-splicing, which means that splicing is aided between two RNA molecules. So the intron of one RNA is able to splice out the exon of another RNA. And the intron per se is released as typically a Y-shaped structure. Why a Y-shaped structure is because in this case, it is not linking with the G of its own. It is in fact linking with the G of another intron. And so you have the other intron attached to this intron. So both the introns are removed, okay? And as the resultant would be that the exon 1 of RNA1 is in a ester bond with the exon 2 of RNA2. So you can see here that a new RNA is being formed where you have exon 1 of RNA1 and exon 2 of RNA2. Such a phenomenon, it is what is called as trans-splicing. Now, let us look at the spliceosome machinery. That is the machinery that carries out these two transesterification reactions to splice out the introns. So, effectively, the spliceosome machinery is made up of certain small nuclear RNAs. As you can see in this picture over here, this is a picture of U1 snRNA. Okay. Now, this U1 snRNA you can say has a typical secondary structure. Likewise, U2 snRNA, U4, U5 and the U6 snRNA, which are part of the spliceosome machinery, all have such typical secondary structures present. And the secondary structure of the RNA is essentially the one that is responsible for splicing. So in this, the RNA molecule play essentially a very important role in splicing. But the spliceosome machinery also has various different proteins. Almost 150 odd proteins are present, but these proteins help in the process of the splicing machinery, albeit the fact that they are helping the RNA to carry out, the small nuclear RNA to carry out the splicing. So when U1 snRNA is bound to by proteins, the U1 snRNA becomes stabilized because of its interaction with proteins. And together they form what is called as ribonucleoprotein, and so this is called as U1 SNRP. You know, another very popular example of a ribonucleoprotein is the ribosomes themselves. Of course, these SNRPs are much smaller compared to the ribosomes, but they are examples of ribonucleoproteins. So similarly, when all of these bind to proteins, you will have U2 SNRP, U4 SNRP, U5 SNRP, and the U6 SNRP. So SNRP is basically an abbreviation for small nuclear ribonucleoproteins. Okay. Now these RNA are found to be anywhere between 100 to 300 nucleotides long, not very long. Okay. And they are associated with these proteins to form these SNRPs. Now, how are these useful in the spliceosome machinery? So they basically have three roles. Okay, one is that they would recognize the 5' splice site and the branch point, which are both conserved regions of the intron. So effectively, the SNRPs help in identifying the introns. Number one. Second, they try to bring these conserved regions of the introns close to each other. So they bring these splice site, branch point together in close proximity. So that is the second job. And the third job, of course, is to carry out the transesterification reactions, which is breaking of one ester bond, and that the energy that is released on breaking that ester bond is used to form the other ester bond. So here, effectively, You don't need ATP for the formation of the ester bond, but of course you need ATP for other purposes, like for example conformational changes or RNA protein interactions or protein protein interactions. These require ATP. But the catalytic reaction itself, that is a formation of a new ester bond by breaking an old ester bond, that catalytic reaction does not need an ATP, because the breaking of one of the ester bonds releases enough energy to form the other ester bond. So that is something that is very interesting in the spliceosome machinery. Now let us look at gradually and slowly how the entire process of splicing happens, keeping in mind that the entire mechanism is finally leading to the two transesterification reactions. And so there are several complexes formed which eventually lead to forming the final complex that has the catalytic action. So it's all a regulated mechanism. And secondly, this entire process of splicing is thanks to RNA-RNA interactions, RNA-protein interactions, and protein-protein interactions. So interplay of these interactions are leading to what is called as the process of splicing. So let's just look at the RNA primary transcript, the pre-mRNA basically. It has exon 1, it has exon 2, and of course this is the intron region with the branch point A, with the 5' splice site, with the polypyrimidine tract, and with the 3' splice site. So this is how the intron is uh, demarcating itself from exon 1 and exon 2. Okay. Now, the first spliceosome machinery molecule that comes and binds is what is called as the U1 snRP. Now, this U1 snRP, basically the interaction is with the 5' splice site. Why is it able to specifically bind to the 5' splice site? Because the small nuclear RNA of the U1 snRP has a sequence that is complementary to the 5' splice site. So you can see GUA, AGU, that is the sequence, and you have a complementary sequence of CAU, UCA present in the U1 snRNA. So here what you observe is that the U1 snRP is able to specifically bind to the 5' splice site. So you can in other words say that the U1 snRP is marking the 5' end of the intron. So the border of the exon 1 and the intron at its 5' end is being marked by the U1 snRP. Then what comes in is a second protein which is called as the U2 auxiliary factor. This U2 auxiliary factor has two basic subunits. One is the large subunit at 65 kilodaltons and the other one is the small subunit at 35 kilodaltons. So they bind to the intron in such a way that the U2AF65, that is the larger subunit of U2AF, is actually binding to the polypyrimidine region of the intron. And the U2AF35, it has within its structure, okay, amino acids that specifically can bind to the 3' splice site. That is the AG, conserved AG, can bind to a set of amino acids present in the U2AF35. So here the interaction is not between RNA and RNA, but it is between a protein and the RNA. This U2AF now is able to recruit another protein which is called as branch point binding protein. So effectively, at the end of these three constituents coming and binding to the intron, what is formed is the complex E. But interesting to note is that every conserved region of the intron that speaks out that it is an intron. You see 5' splice site says it is an intron, branch point A says it is an intron, polypyrimidine and the 3' splice site say that it is intron. So these are defining the intron, and these defined points in the introns are all now marked by molecules of the splicing machinery. So in other words, you can say that the complex E is actually nothing but identification of the intron. And so, to repeat what was mentioned earlier, this is a typical RNA-RNA interaction, and a very specific one at that, that helps marking the intron. The second is an RNA protein interaction, which means that the branch point sequence is interacting with the branch point binding protein. But this branch point binding protein is also interacting with the U2AF. So here it is a protein-protein interaction, and here it is an RNA protein interaction. While here, this is typically an RNA protein interaction where the RNA, that is the intron, is interacting with amino acids of the U2AF to form an interaction. So all these conserved sequences that the introns have are being marked by the splicing machinery constituents. This is significant. This is very important as a start point of the splicing mechanism. Now let us go to the next step. What happens next is that this branch point binding protein is replaced by another constituent of the spliceosome machinery, which is the U2 SNRP. Now this U2 SNRP has a U2 snRNA, and a part of the U2 snRNA has a sequence that is complementary to the sequence in the branch of the branch point. So the conserved branch point sequence has complementarity to the RNA that is present in the U2 SNRP, but there is a catch. What is the catch? The catch is that this sequence that is there of the branch point has UAC, UAAC. But the snRNA of the U2 SNRP has a sequence that is complementary to 6 of the nucleotides of the branch point, but there is no complementary sequence for this A. And this A is the highly conserved A. So effectively what happens is that the U2 snRNA interacts with all the 6 nucleotides except the A, which is highly conserved. So effectively this A is accentuated out. So you can see that it is basically like this, right? So this A juts out, okay, because there is no complementarity over here. And this is important. Why is this important? Because the A being accentuated over here leads to its 2' OH, okay, getting ready for nucleophilic attack. So the binding of the U2 SNRP to the branch point ensures that the nucleophile is ready for the first transesterification reaction. So this is effectively what is observed. You have the U1 bound at the 5' splice side, the U2 having coming bound to the polypyrimidine, U2AF, okay, and this U2 binds in such a way that the A is accentuated, and this A is the nucleophile that will carry out the first transesterification reaction. This that is formed is what is called as the complex A. And like mentioned, the 2' OH of the A can now carry out a nucleophilic attack at the 5' splice site of the intron. So let's see what happens next. Now, after you have the A getting accentuated, okay, the U2AF6535 that leaves the intron, and in place of the U2AF65 comes in a tri-SNRP. This tri-SNRP, that means 3 SNRPs, that is U4, U5, U6 as the complex, comes and binds now to this intron. How does it bind? The U6, its snRNA can bind to the branch point RNA and the as well as the U2. So the U6 can now bind not just to the branch point, but it can also bind to the U2. So effectively U6 is forming an interaction with the U2 SNRP. Okay, but what is also interesting is that the same U6 can also interact with the 5' splice site. So when the tri-SNRP comes, it does one major function, and that is because when the tri-SNRP binds to U2 and it can bind to the 5' splice site, it is basically bringing the 5' splice site closer to the branch point A. Why? Because the SNRP Tri-SNRP can bind to both U2 as well as to the U1, correct, and the 5' splice site. So the tri-SNRP is effectively binding to the 5' splice site of the intron and also binding indirectly to the branch point. So the branch point comes very close to the 5' splice site, whereas here you can see is a distance. Okay, that distance has been minimized thanks to the tri-SNRP. Now, this is basically just showing you how the U6 can interact with the U2. So you have typically an RNA-RNA interaction taking place over here. And this that is formed is what is called as the complex B. So you can see how from complex E, one gets complex A. From complex A, now one is getting what is complex B. So you can see sequentially the entire machinery is getting ready for the first transesterification reaction. Now what happens next is that once the tri-SNRP brings the 5' splice site close to the A, the U1 SNRP goes away and the U6 itself remains bound to the 5' splice site because it has a sequence in the RNA that can bind to the 5' splice site of the intron. So effectively what is going to happen is that the U6 on one end is bound to the U2 and on the other end it is binding to the 5' splice site of the intron. So therefore the tri-SNRP is now bound to the 5' splice site and to the branch point and the U1 has been removed. So which means that the 5' splice site is now available for a nucleophilic attack. So this U6 snRNA, okay, that interacts with the conserved 5' splice site replaces the U1 SNRP to prepare for the first transesterification reaction. And the branch point A is in close proximity to enable the nucleophilic attack. So this is what is happening. So to look at it closely again, when you have the U6 interacting with the 5' splice site and it interacting with the U2, okay, bringing the A close to the 5' splice site, the U4 leaves the tri-SNRP because its job was just to bring the tri-SNRP to the intron. Once the necessary interactions have been established, the U4 leaves. So what happens immediately after the U4 leaves is that there is a formation. Therefore, this that is formed is what is called as the complex C. And it is this complex C that is the catalytic activity, or it has the catalytic activity to carry out the first transesterification reaction. So as we all know, the 2' OH through a transesterification reaction will enable the G of the 5' splice site to interact with the A. So you can see that already a lariat has been formed. But the exon 1 has not been released by the tri-SNRP. So this exon 1, which now has a free 3' OH by the U5, okay, is brought closer to the 3' end of the splice site of the intron and to the exon 2. So the U5 SNRP positions the exon 1 in such a way that the 3' OH of the exon 1 can now carry out the second transesterification reaction between the 3' splice site of the intron and the exon. So effectively you will have exon 1 forming a ester bond with the exon 2. So this is what you are going to get, where exon 1 and exon 2 are together while the intron is released as a lariat along with the tri-SNRP. Along with the, sorry, not the tri-SNRP, but the U5, U6 and the U2 snRNP. So therefore, eventually these SNRPs will dissociate from the lariat and the lariat can be broken down. But at the end, what has definitely happened is that exon 1 is now joined with exon 2 and the intron has been spliced out. Now, what ensures that the splicing is correct? So there are two means by which it is ensured. One is, we already know that the alpha CTD tail of the RNA pol plays a very important role in regulating the RNA processing events. So you could see that when 5' cap has to be added, the three enzymes for 5' cap are added to the alpha CTD thanks to the kind of phosphorylation that happens at the serine. With RNA processing, what is observed is, splicing mechanism, what is observed is that the molecules that are required for splicing are bound to the alpha CTD tail when the serine 2 residue is phosphorylated for sure. So therefore, the proteins that are required for splicing come and bind to the alpha CTD tail. And as the transcript is formed, the transcript being made up of mosaics, that is the coding region and the non-coding region. So say this is the intron. So you have the U1 SNRP coming and binding to the 5' splice site, and you have the polypyrimidine track coming and binding to the 3' splice site. So you can already see that as soon as the 3' splice site is formed, the polypyrimidine tract, the U2AF, that is the U2 auxiliary factor, comes and binds to the 3' splice site, while the U1 SNRP comes and binds to the 5' splice site. So this transfer of the proteins, spliceosome proteins from the alpha CTD to the RNA transcript as the transcript is being formed ensures that the 5' splice site and the 3' splice site of the introns are marked by these spliceosome machinery. Another regulation or another means of ensuring that the splicing is correct is by certain cis elements themselves. And these cis elements are present within the exons. So, what are cis elements? Nothing but a sequence of DNA. So, the sequence of DNA that is present in the exons, okay, they are called as exon splicing enhancers. So, these ESC, that is the sequence that is present in most of the exons, okay, are able to bind to proteins that are called as SR proteins. So when these SR proteins bind to these ESC, these SR proteins are able to position the spliceosome molecules close by. So you can see that when you have exon 1, within the exon 1, when you have the ESC present over here, that binds to the SR, and the SR positions the U1 SNRP right at the 5' splice site. So these SR proteins and the exon splicing enhancers also play a role in ensuring that the spliceosome molecules come and bind to the right parts of the intron. So that is how correct splicing is being ensured. Because see, one should ensure that parts of the exons are not cut off while preparing the functional mRNA. So let's make the conclusions. The mosaic of the gene is transcribed into a pre-mRNA from which the introns are spliced out and the exons are joined through two esterification reactions. This process is what is the RNA splicing. The introns have conserved 5' splice site, 3' splice site, and a branch point A, which are hallmarks of the introns that can be recognized by the spliceosome machinery. The spliceosome machinery contains several SNRPs, U1, U2, U4, U5, U6, and other SNRPs as well, but these were the main ones, and of course several proteins, which through RNA-RNA interactions or RNA-protein interactions or even protein-protein interactions identify the conserved regions of the introns and regulate the splicing mechanism. The introns are removed as lariats after both the transesterification reactions and then can be degraded, while the exons are joined together to form the functional mRNA. Thus, RNA splicing is the second RNA processing event which through spliceosome machinery regulates excising out the introns accurately, ensuring that parts of the exons are not spliced out, but the exons join each other to form the final functional mRNA. Thank you.